So thank you very much for the invitation to speak here. I thought it would be helpful to discuss why we think that XMRV is not a human pathogen. I appreciate that a lot of people had high hopes that XMRV might offer a good deal of opportunity for CFS diagnosis and treatment, and that hasn't turned out to be the case. So it's important to consider where we went wrong. So today I'm going to discuss what is XMRV. I'm going to discuss what evidence suggested XMRV was a human pathogen and what evidence suggests that it's not, in fact, a human pathogen. Where did it come from and what have we learned from this story? And can we avoid erroneous association between pathogens and disease in the future? So XMRV began with a study published in a fairly high-impact journal, PLOS Pathogens, from Bob Silverman, Joe DeRisi, and Don Gannam. These guys used virus discovery tools to seek novel pathogens in prostate tumours. And they came up with what is essentially a mouse retrovirus called xenotropic murine leukemia virus. That's what it is. And they called it related virus, so XMRV. So the fact that it was found in human samples suggested it might be a human virus. This finding wasn't treated with a tremendous amount of enthusiasm from the field, partly because the virus wasn't actually found in the tumour cells, it was found in the stromal cells around the tumour, suggesting that it was unlikely to be a cause of the tumour. So there was a sense of let's wait and see what Bob comes up with next. So what are these viruses, these mouse gamma retroviruses? Well, there are two types of human retrovirus. There are the lentiviruses, HIV-1 and HIV-2, and there are the delta viruses, the HTLVs. Gamma retroviruses, which are found in mouse, are over this part of the tree and quite unrelated to the viruses found in humans. And nobody has ever found any gamma retroviruses in humans before. So I'd like to tell you a couple of salient features about retroviruses, what they are. So retroviruses do the same stuff as other viruses. They go into cells, and they replicate, make new viruses, and those new viruses come out and infect new cells. And two important points about retroviruses are that they have an RNA genome, so they have two pieces of RNA inside, and they turn those pieces of RNA into a single piece of DNA by a process called reverse transcription. They then take that piece of DNA and they integrate it into the host chromosome. So they actually open the host DNA, insert their bit of DNA, and then close it up again. And once that is there, we call it a provirus, and that is, you can't get rid of that. A cell can't remove that from its genome. And it is possible for a germ cell to become infected with a retrovirus, like a sperm or an egg. And if that egg or sperm then goes on to fertilize and generate a new individual, then all of the cells of that individual have that retroviral sequence in them, and that's permanently in that individual, and then their offspring. So those sequences are called endogenous viruses, and when the human genome was sequenced, it was found that 10% of the human genome actually is comprised of these retroviral sequences. So considering that 2% of the human genome encodes coding sequences, there's more retrovirus than there is human coding sequence in the human genome, and it's similar for mice. And these sequences have function, they're important for transcriptional control and how the genome is expressed. So an individual who gets infected with a virus in their germline can then spread. And so most of the endogenous sequences that are in our genome have been there for long enough that they're in the same place in all of us. So we weren't really paying much attention to XMRV as retrovirologists, but we certainly started to pay attention when this paper came out from Judy Mikovits and colleagues. And there were two reasons for this, I think. Firstly, it was very clear that ME/CFS is a very difficult disease to diagnose, and therefore its cause is unknown, and therefore really quite difficult to treat. And a viral cause, particularly a retroviral cause, would offer pretty straightforward diagnostics, similar to those used to diagnose HIV, and also similar treatments. The drugs that are used to treat HIV, which are very good drugs, are also, some of them are active, most of them are active, against gamma retroviruses. And there's also the possibility of a vaccination. So that's one series of reasons to take this seriously. But another reason was that this study, and a couple of others, suggested that there was quite a large proportion of the American population and presumably the world's population infected with this virus, but without symptoms. And there was a suggestion that maybe these people would go on to develop prostate cancer or chronic fatigue or something else. So it seemed very important to sort out as quickly as we could whether or not this really was a pretty common new human virus. So pretty quickly the situation became more confusing in that the positive studies kind of slacked off. There were fewer of them, and a series of negative studies started appearing. So people testing for XMRV in patient samples were not finding them. It's quite a confusing situation. So the question was, is XMRV circulating in the human population? So we were aware of this study from Dusty Miller's lab in Seattle. Dusty is a retrovirus aficionado and was pretty trustworthy. Dusty showed that there was a cell line in common circulation, many of us had this cell line in our lab, that appeared to be infected with the XMRV virus. And this was not entirely unexpected, perhaps, because it was a tumour cell line derived from a prostate cancer. So perhaps that reinforced the idea that this was a prostate cancer-associated virus. However, we were very much aware of the fact as to how these type of cell lines are generated. So if you take a prostate tumour from somebody and you put it into cell culture in the lab, it doesn't tend to grow, it tends to die. But if you take that tumour and you put it into a mouse, and then you wait till the tumour grows, and then you sacrifice the mouse and then put it into another mouse, and you do that several times, then the tumour acquires the characteristics of being able to divide continuously, and you can end up with a cell line, which is a very useful tool. So this had been how the 22RV1 cell line was generated. And during this process, we also knew that it was quite common for these cell lines to pick up mouse retroviruses. A particular class of mouse retroviruses, xenotropic viruses, were common in this regard because they're very good at infecting human cells in the lab. So many of the cell lines that passage through mice are infected invisibly with these xenotropic viruses, and you only see them if you use virus detection techniques to take a look. And the 22RV1 cell lines had a xeno in them, this XMRV clone. We were also very much aware of how retroviruses can be erroneously associated with disease. David Griffiths was a PhD student in the lab next door to me when I was a PhD student, and he spent 5 years working on a virus that he believed was associated with certain autoimmune diseases. However, after 5 years, his postdoc demonstrated, the two of them demonstrated, that in fact the virus was an endogenous sequence in rabbits. So his samples had apparently been contaminated with rabbit DNA. We didn't have a lot of rabbit reagents in the lab at the time, but we had rabbit antibodies, and we assumed that rabbit DNA could find its way into the samples, and using extremely sensitive detection techniques could lead you to believe that you had rabbit virus in your samples. But it turned out that that was simply contamination from rabbit DNA, so we were very much aware of that as a possibility. So the way we approached to determine whether or not XMRV is spreading in the human population was to compare the sequences of the DNA of the virus in the cell line to the sequences that had been produced by Judy and by Bob from the chronic fatigue and prostate cancer patients, respectively. So we use phylogenetics to do this. This can get quite complicated, but essentially we took all of the gamma retroviral sequences we could find and put them in a software that compares their relatedness to each other, and you find they go into the three families of retroviruses, the polytropics, the modified polytropics, and the xenos. And we found that all of the XMRV sequences clustered together as you'd expect. We found there was very little variation between them, which is a surprise. But the most important point is that the blue sequences from the cell line intermingle with the red and orange sequences from the patients. And you would not expect that if they had separate sources. If the blue sequences were simply an example of a prostate cancer that had XMRV, it would be expected to form its own cluster. But in fact, they intermingled, suggesting that they were all coming from the same source. What is that source? Well, the fact that the blue squares are at this position in the tree tells you that these are the origin of the other sequences. So they are parental to the other sequences. So another way to look at that, perhaps simpler, is the what we call a genetic network. So if we look at all the sequences, so we're looking at a 1,663 base pair piece of DNA, and 4 of the clones have exactly the same sequence. All of these guys, all the blue ones from the cell line, have a single difference. And then we have the patient sequences. And most importantly, you can see that one of the sequences from the cell line has a change, A178G, and all of these guys from the patients also have that same exact change, but then some of them have an additional 1, some of them have an additional 2. So that very, very strongly suggests that this clone is the source of those, and this clone comes from the cell line. So that was essentially the bottom line as to what made us make the assertion that XMRV likely came from this cell line. So another key piece of evidence that it was a human virus was the fact that retroviruses integrate, and Sam Chow's lab had shown that if he sequenced the junction between the virus and the human genome, he could find in patients the junction that's human, 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 virus, 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 human, human, human. And that was very strong evidence that these samples did represent infections in human samples because you could find the junction. However, we felt that was likely due to contamination because we also worked out that Sam was infecting cells in the lab at the same time that he was amplifying samples from the prostate tumors. So we compared his list of 14 samples from the tumors, 14 junctions, with 472 junctions from the cell line, and we found that 2 of 4 of them were identical. So you can see here that the upper and the lower line are the same. So that would tell you that despite the size, the enormous size of the human genome, 3 gigabytes of choice, the where to integrate, on 2 out of 14 occasions they'd gone into the same place. That seemed very unlikely, and again undermined the notion of XMRV being a human pathogen. And to his credit, Sam went on to show that, once we published our paper, Sam went on to show that indeed he could show that he had inadvertently contaminated his prostate cancer samples with his cell line samples. So what we really wanted to do was to find out whether the tumor that gave rise to the 22RV1 cell line had the virus in it before it went into the mouse. So in order to do that, you'd have to have a sample from the tumor before it went into the mouse, obviously. We made some calls to Cleveland where these experiments were done, and we didn't get anywhere. But it turns out that a friend of ours, Vinny Patak, who's an investigator at the National Cancer Institute in the United States, Vinny's brother-in-law happened to know somebody who worked in the lab where those experiments were done. So Vinny got his brother-in-law to call up his mates and, and send him the samples that we all wanted. That allowed Vinny then to go on and test whether the tumor had the virus in it beforehand. So the DNA samples were available from early passage. In fact, they weren't available from before it went into the mouse, but they were available in the early mouse experiments, and there were some samples available from the later mouse experiments. Vinny could show by using PCR that the XMRV virus could not be found in the early passage, but was found in the late passage. So that tells you that that virus infected those cells on a particular date at the time that those experiments were going on. But more importantly than that, Vinny was able to show that XMRV is what we call a recombinant, which is a mixture between 2 other parental viruses. So Vinny identified 2 viruses, which he called pre-XMRV1 in blue and pre-XMRV2 in green. And he could show that the XMRV virus was a mixture of these 2 viruses. And if you remember earlier, I told you that the virus has 2 RNA genomes, so it's possible for a virus to be formed from one of these and one of these if both of these sequences are in the same cell. And then during the process of reverse transcription, the virus jumps between these 2 strands to make its DNA molecule, leading to a new virus referred to as a recombinant. So the chances of those switches occurring at those specific 6 points on 2 occasions, Vinny calculated to be 10 to the 12. I'm not sure about Vinny's maths in that regard and how he worked that out, but it's certainly a very large number. So the bottom line is that not only did the virus infect the cells, the cell line at that time, but it was also generated at that time because he could show that XMRV was not in the mouse prior to this experiment being done. He could only find the parental viruses. So finally, I think the final closure of this story is Ian Lipkin's study, where he took 150 CFS patients and 150 controls, and then sent those samples, blinded, to the labs that had previously claimed to be able to detect XMRV in patient samples. So that was Xiaoqing Lo at the FDA and the Mikovits-Rossetti collaboration. These guys did not detect any virus in any of the samples using their PCR detection techniques in this case with these new samples. Frank Rossetti found some positives with plasma, but the fact that he couldn't distinguish between the controls and the CFS cases suggests that that is non-specific interactions. So I think that study particularly really puts this story to bed. We can now say that the virus in the patients that were the clones generated by Bob Silverman and Judy Mikovits were from the cell line, and the cell line got the virus from the mouse, and the mouse generated that virus on a specific date in 1994. So that could not then be the cause of CFS ME. So the genetic evidence really contradicts the notion that XMRV is a human pathogen. The XMRV integration sites were shown to be on a case of unfortunate contamination, and XMRV is a recombinant with a specific birth date. So what have we learned? I think what we've learned is that PCR strategies have to manage the inevitable level of contamination that you're going to generate if you use very sensitive techniques. I think there's been, particularly in the early part of this work, there was a sense that you must manage things such that you don't get PCR contamination, and I think that's probably wrong. I think you need to manage it in such a way that you understand the difference between PCR contamination and an actual virus, and you should be able to do that using genetics. It's also very important to understand the sensitivity of the PCRs. We didn't really think that these could detect single molecules floating around in the air, but it turns out that they can. So you cannot make DNA, plasmid DNA, in your lab and then sensitively detect the same sequence and expect to be able to distinguish the background you generate from making that plasmid in your lab and what you might find in a patient. So I think we've learned a good deal about how to manage PCR sensitivity. I think we've also learned that genetic evidence really can be used to determine the origin. Phylogenetics is a very powerful technology that I think is underappreciated generally in science. And I think that we need to accept the fact that if we're going to assign a new pathogen to a disease, we need to do it in a rigorous way. Colleagues of mine, Paul Kellam particularly, at the Sanger Institute, has been through this on a number of occasions. I won't go through it, but this is an article where he discusses the kind of rigors that are required to assign new pathogens. So thank you very much for your attention. None of this work was really done in my lab. It was done in collaboration, particularly with Stefan Huet, who's here today. I would be happy to answer any questions about viral genetics with Jeremy, Eleanor, Deenan, and Tan. We did the phylogenetics work with Eris Katsarakis and Olivier Pybus in Oxford. We collaborated with Paul at the Sanger Institute. The XMRV mouse work was done in Vinny's lab in collaboration with John Coffin by Toby and Oya. And I'm a senior fellow at the Wellcome Trust. So thank you very much.
Thank you, Greg. Well, that's clearly a very nice piece of detective work, which clarifies a lot of the mystery that surrounded a lot of, high prestigious work. Has anybody got a question? I mean, I have one. Is it possible also to mix cell lines up as well? I seem to remember in my time, people mixed up HeLa cell lines with some others. I mean, you've got to be very careful in this field generally to identify what you're working with.
That's absolutely right. We have in the past found cell lines that are, not what you think they are. They're not the same species even. And, I think you have to be on top of that.